Well, hello, AP Bio students, and I just wanted to give you a starting point for Tuesday. We're going to go over the little tiny bit of glycolysis left and then Krebs cycle. And then there's going to be a second video that I would like you to watch about the electron transport system. And like I said in previous videos, this should give us a nice head start so we can then dig in and practice and review on Tuesday. On Friday, and by the way, you have these notes, which you should definitely get back out again. And I just am giving you some extra pictures. You do not need to try to copy them down. This is just for you to be able to see it. So on Friday, we did the general overview of cell respiration and then glycolysis. And I gave you a very simple version of it. So I had mentioned in class that the energy levels in glycolysis, it's like you have to jumpstart glucose in order to then get everything, and that's absolutely true. So I did pull off a graph that actually shows the energy levels, and you can see right about here we're boosting it up in energy. We're boosting it up in energy, and that's going to take 2 ATP to do that. But notice we've raised it up enough that it falls back down. That's where we're going to get our 2 ATPs from. And then we get another drop and we can get 2 more ATP from. So what you want to imagine, because sometimes it's a little abstract for students to think, well, we started with 2 ATP, but we can get 4. Like what happened there? Well, it has a lot to do with the energy that's in glucose, that as we break it down, we can get that from all of the glucose, was this big broken down. So now the other thing I gave you was a little outline of glycolysis that was a little bit more detail. And I do not mean to imply that you have to know this in detail. So the idea of glycolysis is you want to take away the main ideas. There's an energy investment to be that needs to happen, and then there's an energy um, release that we can then get our ATP molecules from. So notice that we're going to add these 2 ATPs, and I had a similar picture. I get it's not glucose but fructose at this point, but we're adding 2 phosphates. That really kind of contributes to that increase in potential energy. And then we're going to get down to a 3-carbon sugar called glyceraldehyde, and then that is going to go down to pyruvate. So there's one diagram, and then you have one that's kind of similar that I gave you that shows the details. Now, here we have glucose, and notice with each one of these there's a different enzyme: hexokinase, glucoisomerase, phosphofructinase, aldolase, and all of these enzymes are going to be involved in manipulating and breaking bonds. So for instance, we can add a phosphate here, we can add another phosphate, that's where we're getting our fructose, and we have boosted that energy. And then the second phase, this is going to be our energy payoff. And at this point, we have a dehydrogenase, a dehydrogen. We're removing hydrogen, and we're going to get our NADH. And so there's our high energy electrons. The transfer of the hydrogens and the electrons, they don't lose a lot of potential energy that way. And so it really is a little dump truck of a high energy electron. And then same thing over here, and over here we can get our ATPs directly from those molecules. Now this is called, and you do this one, you want to jot down. I know you don't have the picture, but you can jot this down. The ATP that we get from glycolysis is through a process called substrate level phosphorylation. Now remember, all phosphorylation means is that we're putting a phosphate on something. And substrate should remind you of enzymes. So here we have our enzyme. And let's put a little, let's put a substrate in here, and let's say that that substrate is our bisphosphoglycerate, and it has that those phosphates on it. So what we want to do then is we want to take a DP, and we want to take the phosphate off of that substrate, that 1,3-bisphosphoglycerate, and that enzyme has that ability to just move this over. And so instead of getting the phosphate from the surrounding areas, we're getting it off of a substrate, and that's where the name comes from. And you can see this happens a couple of times to grab the phosphates that are there. So, and that's where we're going to get our 4 ATPs from it. Now, at this point, we've been in the cytoplasm. So glycolysis occurs in the cytoplasm, but we want to move into the mitochondria. And when it's there, it's going to go through a little conversion called a conversion to acetyl-CoA. So we start off with pyruvate, this 3-carbon molecule, and this carbon is already oxidized. It's had its hydrogen electrons removed, so we remove that as carbon dioxide. We are going to be able to pick up another energy-filled electron and make NADH. And just so you know, when we do that, it started off as NAD with a positive charge. It didn't just come out of nowhere. It picked up a high-energy electron, and 2 of them, and then a hydrogen. And then this is another boost of potential energy. There's this coenzyme A, and we're going to attach it to this acetyl group. So we end up getting acetyl-CoA. And this is a boost of potential energy. And you want to almost think of it as like a little kick to get into the Krebs cycle. So this is the Krebs cycle, and it's also known as the citric acid cycle. And you want to take away the main points. What we're doing in the citric acid cycle is nibbling at these molecules and pulling off electrons and hydrogens that have a lot of potential energy in them. So some books have called this like a slow burn, a slow oxidation. Instead of putting this to fire and unoxidating it very fast, we're kind of slowly taking away those electrons and hydrogens, and that's going to be still going to be oxidation, just very slow. So the nuts and bolts of this, the things you want to take away is that we're stockpiling these high energy electrons. And so let's look at what's happening. We're going to remove 2 carbon dioxide, and we're going to get 3 NADH, 1 ATP, and 1 FADH2, which is just another version of NADH. It's another dump truck. But one thing you got to remember is we got 2 pyruvates. So whatever happens to one pyruvate is going to happen because both of them go to Krebs. So everything we need to do twice. So let's do a quick accounting right now. Accounting of everything. Now in glycolysis, we were able to get 2 ATP, that's net, and then 2 NADH. Now, not a lot of ATP yet. And then we have our Krebs, or citric acid cycle. And here's where you're going to start noticing the numbers climbing. 2 ATP, because we had 1 down here, we had to multiply it by 2. We have 3 NADH here, 1 NADH there. And this, so we can say Krebs, and this is the conversion as well. So we have 8 NADH. We've got 2 carbon dioxide, 1 carbon dioxide. That's 3. Multiply by 2, 6 carbon dioxide. And then 1 FADH2. Multiply that by 2. There's our accounting. So far, we haven't seen a lot of ATP. 2 there, 2 there, not a lot. But the big thing is going to be the electron transport system that's going to be happening. So let's real quickly go over the little details that happen. And again, we don't need to know these in detail, but it is kind of neat so it doesn't become so abstract to you. Because just saying things like, oh, we got 3 NADH, well, where in the world is that coming from? So let's do this in a little bit more detail. We start with that 2-carbon acetyl group, and that's going to join to citric acid. So citric acid is the beginning molecule. It's 4-carbon, and there's going to be enzyme that is going to catalyze this reaction, and we're going to get a 6-carbon molecule. Now, don't worry about the names. The take home is that there is an enzyme that's catalyzing it, and we're combining this. We've now boosted that potential energy again. Now, the nice thing your book does is it color codes it. So notice this is the acetyl group that came in. Now, through this whole cycle, think again about that nibbling. We're nibbling at these hydrogens. So I wanted to show you a couple of them where it's a little bit more obvious what's happening. So notice we have 2 red and then a blue and a blue. Well, if we go down 1, notice this is now gone, and it left as a carbon dioxide. And if we notice further, this hydrogen is gone and this hydrogen, and we've rearranged it to a double bond. So that's where our NADH came from. So we had, again, NAD plus that was able to swing around, pick up 2 electrons, a hydrogen, and that's where we're picking it up, and then putting it in NADH. And again, we can do one more turn where we can see this blue has disappeared, and that disappears as carbon dioxide. And then we bring in another CoA, coenzyme A. We can get NADH from this as well, and you can look and see where we lost that. And you know, we lose the electrons, and they get rearranged from that. And then we can pull off another switch. Now, this they've not put in all the details because we add a phosphate as the coenzyme disappears, and then that phosphate gets transferred to make ATP. So I just wanted to give you a little idea of the actual events, not so that you can suddenly have to memorize it, but that you can at least see that this is indeed happening and where it was happening. So I hope that helps us start thinking about what's going to happen next. So just keep this in mind, the accounting. What we're going to really see is the big payoff for these. So happy rest of the weekend.